they were interested in understand, how we can use some of these tools to study the formation, assembly, and disassembly, and the influence of this biomolecular condensate on membrane. So how the wetting of a biomolecular condensate can affect the membrane and vice versa. And well, spectroscopy and microscopy is a powerful tool for that. So, but before to jump into the result, I want to give a brief of an introduction of what was the topic for them. So they were interested in understanding these organelles that has, appeared or starting to grab attention from the biological community, which are the membraneless organelles. These membraneless organelles are, transient proteins and RNA that can form in the cytoplasma as well as in the nucleus. And regarding different signaling like stress or some others, this can, drive or, move cells to different kind of pathologies. So how this happens is, well, it's triggered basically for changes in temperature, pH, concentration, binding partners like RNA, as I mentioned, DNA. So there are proteins that are, multivalent, so they can interact with many different, other molecules. At some condition, what we have learned is that this molecule coalesce, and this can entrap, a signaling, or can, remove a signal to get into this place, etc. So these are having a lot of attention, and so far we learned that these are very important for neurodegenerative disease as well as for cancer. But why is important of the wetting between these condensate, which are I insist membraneless, so there is no boundary in the surface, with the membrane-bound organelles? Well, because in the cell is well known that everything is together, right? You have proteins, you have lipids, etc. And this group has been working very, very hard in understanding how the wetting can be influencing the membrane dynamics, and in particular, in time junctions, in autophagy, stress granule transport, T cell, and ER interaction with many protein, virus protein. It's well known that the protein can interact with membrane, and there are influence in each other. So the influence of the biomolecular condensate in the membrane as well as the membrane in the condensate. So the question was, well, can we study the effect of condensate upon membranes? Well, yes, we can study. And the tool that we use is the giant unilamellar vesicles. These are, membranes that can grow at the size of micrometer, 10, 20, 50 micrometers of diameter. And then we have a particular system that I will mention in a minute that form condensate depending on ionic strength, these different physicochemical parameters. And we wanted to put together and see how this tool of spectroscopy can help. So this is the model that they use, glycine, and glycine undergoes to biomolecular condensate, and this change in the shape depending on the ionic strength. Sodium chloride can drive this condensated, and they study they did this previous study of how much is the the formation of biomolecular condensate over a long range of concentration of sodium chloride, and they recognize that from 50 to 200 millimolar of sodium chloride, this produce biomolecular condensated. And then they study using biophysical tools. So basically the measurement on the angle in the different, interaction between the condensate with the membranes. And using this approach, they can measure all the biophysical parameters, and these geometric factors can be calculated from images like this. And you can see that from low concentration to high concentration, the weighting on the membrane change a lot indeed. And they measure, these parameters, and they can obtain biophysical information about this process. But there was no information about, what was happening on the membrane, physical parameters of what was happening on the membrane parameter. and this was part of the main goal of this project. So they did also some very interesting studies in which, you know, they can study this wetting very carefully with time resolution and even increasing resolution with step, and they can see that this, you know, webbling or bubbling that the membrane suffer, or ruffling that the membrane suffer here, and has, you know, very dramatic change in architecture of the membrane. So membrane are very sensitive to molecular parameters that can affect hydration, you know, electrophilic, you know, interaction in the surface of the membrane, etc. And this basically was the information they have when they approached to me to help them in the use of this set of probes that are very valuable for study the membrane biophysics as well as the biophysics of the interior of the biomolecular concepts. As I present in my talk of spectral phasor, I have been using, you know, this kind of proof, the dimethylamino naphthalene proofs, to study, you know, in cuvettes, the dynamics of membranes, in cellular, in vivo, you know, the dynamics of organelles, and then, you know, some other methods to combine, you know, fluorescent lifetime with spectral, and then, you know, understanding, oh, again, this computer, oh, sorry. Well, I was saying that we were using these tools to basically understand the interior of the cell, and this is work done with ACDAN, and then we use this to understand also trying to understand the lens and how the lens, you know, was maturating over time, and how was the effect of the mutation of some water transporter. These are aquaporins, etc. So if there are some of you interested in this, please, I will be happy to share some of this literature. But just to remember what I already said the other day about these fluorescent proofs. These are very interesting fluorescent proofs because when they got excited, they enlarge the dipole moment, and the dipole moment can be used to sense the pole active molecule around it. And in particularly, if you change, you know, this R change here, you can give traction for membrane, or you can have a soluble. So basically a dye that can explore the intracellular interior, or a dye that can label to membrane. And in this, if this is the case, anyway, so these dyes are really fantastic because you can use it as a molecular ruler of what is going on in terms of molecular crowding inside the cell, and how water can impact, you know, in different processes. And to analyze this data, basically we use the phasors. We use the FLIM and the spectral phasor because we collect both information. So the first thing that I want to show you is, well, we start with basically calibrating how the ACDAN photophysics change in regards of the different amount of glycine in water. And you can see in this spectral phasor plot of, you know, glycine increasing concentration from 0 milligram per milliliter to 20 milligram per milliliter, that the glycidin is moving backward in the phasor, telling you that it's decreasing, you know, the polarity around the dye. So in a way, it's decreasing the activity of the water. And if we measure this with one of the tools that the phasors enable you to use, like you know, the center of mass of, you know, the linear combination between 2 points, as I showed you yesterday in the practical session, we can quantify this, and then we can plot this over time. And we use this as a, you know, ruler of how much the changes in the concentration of glycidin can affect the photophysics of the probe, and how much is changing the water dynamics in size of these aggregates. So the first result that I want to show you is, well, we already knew that, you know, if we change the concentration of sodium chloride over a long range from 0 to 400 millimolar, the protein will behave, you know, different. And in certain range, we will have liquid-liquid phase separation. And as you can see here, from 50 to 200, as they described before, there was formation of condensate. And the question was, can we understand what is going on inside of this condensate? What is the water dynamic inside of this? And this tool works very, very, very good, because if you look at here, using the same, ruler that we place here, we can now use a color scale in order to show that from low relaxation to high relaxation, while the protein is not condensated, you have very high dynamic of water. As soon as these start to have condensated, you start to decrease the dynamic of water inside the condensate. And this is not just on and off. You have degrees, and you have also heterogeneity in the distribution of the condensated inside. So basically, the tool is telling us that there is a strong heterogeneity inside of this condensated, and also is depending on the ionic strength that you are applying to your system. So we measure this, and we quantify this using the same approach. So we measure between 2 points, that was our calibration, and then we measure the distribution of pixels found between these 2 points, and then we calculate the center of mass of this distribution, and we recover basically the same profile that was shown before with a different technique. So we basically define a new phase diagram for this condensate, but now based on the physicochemical parameter that we measure inside of the condensate, which is the water dynamic, okay? And the next question that we wanted to answer was, okay, we know that if we put together membranes with these condensates, the condensate will wet the membrane. And if we change some physicochemical parameter like ionic strength or some others, it will change the condensate. It is true that the condensate will also affect membrane dynamics. So this was the question that we want to answer. So instead of using Agdan to measure, the condensate, which was what we just did before, now we use Laurdan to label the membrane, and then we use condensate that were attaching to the membrane. So this is the experiment here, and Laurdan, as I explained before, it sends, the change in the packing of the membrane, so the organization, the physical parameters of the membrane. And we can measure this, and you see that, if we look at the distribution that we got for the Laurdan, and we place this cursor here, and we place this trajectory in order to measure how it's changing the membrane, we realize immediately that the membrane that was wetted by the condensate was different than the membrane that was not in contact with the condensate. And this is basically this shift that we can see here in the cluster, in which the more pink here is the part that is attached in contact with the condensate, and the more cyan here is the one that is remain not in contact with the condensate. And I want to make a point here. These giant unipolar vesicles were built only with one phospholipid, diolioyl phosphatidyl-choline, which is fluid at the temperature of experiment. So the phase transition of DOPC is below 0 degree. So it's totally fluid. I couldn't believe that this was happening, because, it's a really, really, really fluid membrane that by the attachment of this condensate can modify the physical parameters of this lipid. And this was really an impact for me. I couldn't believe that this happened, but well, the measurement tell us that this is true. And then we will start to measure what was the effect if we modify the physicochemical parameters that we knew will affect the condensate. It will affect the way that the condensate wet the membrane, how much this may impact on the membrane, right? And we did the same experiment than before, but now changing the sodium chloride and measuring, this fluid diffraction we call, we can identify, profile, and these shadows here are the error that we have. And basically we did this several times in this paper with, many controls, etc. And we can measure, again, the center of mass of this distribution, and you can immediately notice that as soon as we, increase the concentration of sodium chloride, it also changed the fluidity fraction of the membrane. So the membrane get more and more and more order in this region where it's sticking to the membrane. So the next question was immediately, on front of eyeglass. There is an effect on the other left membrane that is not in contact? And the answer is no, at least with the sensitivity we have. There is a very little trend that we cannot assign because the error is very big. So basically, the wetting is modifying the dynamic of the membrane, which is in contact, but it's not producing a strong effect in the left CUV, which is not in the with the condensate. Sorry. So this was part of this paper. We then did a lot of experiment with other condensate because this is a protein, but we wanted to generalize this using a more general principle, and we use a mixture of PEG with some other crowder, and if you blend these two crowders at certain amount, you are able to produce condensate inside CUVs and outside CUVs, and you can modify this. And we use film and you use spectra. So if you want to go through the paper, you can take a look of this. But basically, we end up proposing something new, which was, well, the effect of the condensate of the membrane is a fact, but also the environment that is where is the condensate can also trigger effect on the membrane, which is something interesting because now we can come back of what is the purpose of this condensate. The purpose of condensate is to produce, an entrapping of some signal or something like that in the cell. But this condensate can also affect signaling in membrane. So in a way, you can go from one organelle to another, or from intracellular to extracellular, translating signaling due to physical changes in these structures. And basically, they are now convinced that this can also be very powerful when you don't have just a simple membrane. You have many lipids in the cell, and you have cholesterol, and you have other things in the membrane like proteins. So they are now pushing further in this area of research, and I like it to use this example because it's very nice how powerful can be these techniques to understand biology at the molecular level in an in vivo experiment, right? Or not in vivo, but basically in real time and space experiment, right? And how relevant it is for biology? Well, this protein is related to the functionality of the tonoplasm in plants. This is a plant protein, and there was evidence that this condensate can attach to vacuola in this plant, and now they have a physical support that this was doing something. This was changing the building of the cell because they wanted to produce a signaling or something else in the cell. So with that, I want to thanks to Agustin Mancharotti. Agustin Mancharotti was the leader, of this project in Germany, working in the Rumiana di Mova. And I also want to thanks, all the sponsors that usually help to have this experiment. And I hope this very crashed talk still convince you that it's a useful technique for understanding biology. Thank you.